Hello. Okay, so this lecture is going to be part 3 of our chapter 9 covering muscles and muscle tissue. There's not very many slides left, so this should be a rather short lecture here, but I wanted to just break it up a little bit so you weren't watching lectures that were over an hour long. Okay, so in the previous lectures, we talked about just like muscle anatomy, mostly microscopic anatomy. And then part 2, we talked about the muscle physiology, so what exactly is happening on a cellular level to cause muscles to contract. So from the neuromuscular junction, end plate potentials, excitation, contraction, coupling, and then the cross bridge formation and cycle there. So those were looking at what's happening like in an individual muscle fiber. The rest of this is really talking about kind of a bigger picture. So what's happening in the whole muscle is really happening. What is happening in the fiber is happening in the whole muscle as well. Okay, so when the muscle contracts, we have multiple fibers contracting at the same time, which is going to be producing muscle tension, okay? And that's the force that's going to be exerted on a load or the object to be moved, okay? And that's really just a result of the contraction from our individual muscle fibers, but together we call that muscle tension when it's on a bigger picture, okay? Contractions may or may not shorten the muscle itself, which is kind of a little bit counterintuitive. But we can have what's called isometric contractions, where you don't have shortening. So muscle tension increases, but does not exceed the load. So if you're just holding something in front of you, you're not contracting the muscles to shorten them. You're just holding it still, but still maintaining tension in your muscle or increasing tension in your muscle, but you're not like actually contracting, right? So if you're holding a plank, you're increasing muscle tension, but not actually contracting those muscles to shorten them to move. Whereas isotonic contractions, the muscle actually shortens because the muscle tension is going to exceed the load. So if you're doing a sit-up or a push-up, right, you're actually shortening your muscles shorten your muscles, and it's going to exceed the load, so that when you do a sit-up, your contraction is going to be greater than just the force of, your body weight pulling you down because you want to sit all the way up. Okay, if that makes sense. So isometric, there's no shortening. The tension increases, but isn't exceeding the load of whatever is putting on your muscle. Isotonic, the muscle actually shortens. Okay. The force and duration of contraction will vary in response to stimuli of different frequencies and intensities. Okay, so it depends on what the stimuli is. Depending on what the stimuli is, we'll have different types, forces and durations of muscle contractions. Each muscle is served by at least one motor nerve. Okay, remember the motor nerve contains axons up to hundreds of motor neurons. Axons are going to branch into the terminals, each of which forms one neuromuscular junction with a single muscle fiber, but you can have axons branching to form multiple terminals into multiple muscle fibers. And we call that the motor unit. So the motor unit is the nerve muscle functional unit. Okay? So, again, the motor unit consists of the motor neuron and all of the muscle fibers that it supplies. So some only supply like 4 muscle fibers, some supply up to 100 muscle fibers. The smaller the fiber number, the greater the fine control. So in places that we have really fine control, like our fingers, one motor neuron is only going to innervate a few muscle fibers so that we can have really fine-tuned control. Okay, we have some that each motor neuron is going to innervate hundreds of muscle fibers. That's going to be areas in like a quadriceps where you don't have fine controls of all the little muscles, but you have one muscle with a lot of muscle fibers that you want to contract at the same time to do the same thing. Okay. muscle fibers from a motor unit are spread throughout the whole muscle, so stimulation of a single motor unit causes only a weak contraction of the entire muscle. Okay, so anytime you have an action potential coming down the axon of a motor unit, obviously it's going to go to each and every one of these axon terminals. Okay, and these axon terminals are going to be spread throughout the muscle to cause contraction in each individual sarcolemma, but you have, action potential coming down to some other muscle fibers as well. Okay, so again, if one motor unit innervates more muscle fibers, is going to be less control. If you have a motor unit that only innervates, this one was only innervating 2, you're going to have a finer control of movement. Okay. We can measure this by looking at a muscle twitch. Okay, and normally in lab you would do an activity where you can see this, but since the lab's online, you'll be just looking at kind of examples of this. But a muscle twitch is the simplest contraction resulting from a muscle fiber's response to a single action potential from a motor neuron. Okay, so if we're measuring the contraction and relaxation of a muscle on a gross level, so not looking microscopically, the Z lines moving together, but looking at the whole muscle and the force that it's contracting, that's looking at the muscle twitch. Okay, so the muscle fiber you're going to see contracts quickly and then it's going to relax. And we can measure this using something called a myogram. Okay, so this here on the bottom is a myogram. And it shows the different phases of a muscle twitch. And in lab, we would, again, normally do an activity where you hook up your pinky to what's called a force transducer. That looks like a little noodle. It's attached to your pinky. And when your pinky moves, it causes that force transducer to move, and that measures the strength of your muscle contraction. Okay. So there's different parts of this myogram that we can look at, and they tell us different things about what's actually happening in the muscle on a chemical level. Okay, so there's 3 phases. We have latent period, and these are the events of the excitation-contraction coupling. So from the action potential, the stimulus being sent, till before any contraction actually happens. Remember, there was a lot that had to happen. So we had the action potential come down, calcium come into the axon terminal, acetylCoA release. AcetylCoA binds to the ligand-gated receptors on the muscle. Those open, allow sodium in. The sodium goes and excites the voltage-gated channels. You have the propagation of the action potential down the cell membrane of the muscle cell, goes down the tubule, releases calcium from the sarcoplasmic reticulum, that goes and binds to troponin, tropomyosin, moves out of the way. All of that happens during the latent period. Okay? Period of contraction is when we have cross bridges starting to form. Okay, when the cross bridge is starting to form, remember we have the power stroke, which is causing actin and myosin to go over top of each other, which is increasing the tension. So during contraction, this phase here, we see an increase of tension, increase of that contraction of the muscle. After the muscle is done contracting, then we have the period of relaxation, which I guess I can do a different color. The period of relaxation is this over here. And that is happening when calcium goes back into the sarcoplasmic reticulum, right? And you have actin and myosin breaking apart, the muscle relaxing. Okay, and the tension is going to go all the way back to 0. And you'll see that the relaxation phase is a lot longer than the contraction phase, which is because calcium needs to go back into the sarcoplasmic reticulum. We talked about how that uses energy, ATP, to pump calcium out of the muscle into the sarcoplasmic reticulum. This is an active process, so it's going to take longer than during contraction. We just have those cross bridges forming and breaking, forming and breaking. So contraction is going to be relatively quick. Relaxation is going to be slower. It's going to take longer for that calcium to be pumped all the way out, for all of it to be pumped out, so that the muscle can fully relax. Okay? So those are the phases, again, of this myogram. of a single muscle twitch. All right. We can have differences again in the strength and duration of twitches due to variations in the muscle, so of different metabolic properties and enzymes between different muscles. So some muscles we want to have really quick, rapid contraction. So muscles of our eyes where we're moving, we want those to be very quick. Whereas larger muscles, like the muscles of our calf, our gastrocnemius, are going to contract more slowly and hold it longer. Okay, so you can see the lateral rectus is a muscle of our eye. We have really fast, short twitches. Muscles of our calf, our gastrocnemius, is a little bit slower, but still pretty quick, where the muscle of our soleus is even slower. Okay, and it's just different variations of the muscles that want to, contract quickly or slowly depending on the function of that muscle. Okay. Normal muscle contractions are relatively smooth, and the strength varies with the needs of contraction. Okay, so that muscle twitch that we just looked at in the myogram is only really seen in like a lab setting or in an individual if they have some neuromuscular problems, but you're not going to see it in a normal muscle. You're not going to see just one single twitch and the muscle going back to normal. You're going to see different graded muscle responses. Okay, so they're going to vary in strength of contraction depending on different demands. Okay, and this is important because it allows for proper control of the skeletal movement, right? We want to make sure that when we're contracting our biceps, we're contracting it in a way that's allowing us to do what we're intending to do. Okay, and these graded responses are graded or changed, varying in strength, depending on the frequency of stimulation and the strength of stimulation. Okay. So we see here the single stimulus results in a single contractile response, which is that muscle twitch we just talked about. Okay, so you need a stimulus, you have contraction, relaxation. Another stimulus, contraction, relaxation. And remember, if I zoom in here, if we draw a line up from the stimulus, you should see a little bit of time. This graph kind of does a shit job of showing it, but the latent phase is the time between the stimulus and when contraction actually starts. Okay. But again, if we change the frequency of the stimulus, we can change the response of our muscles. Okay, so if we increase the frequency of stimulus, I'm going to write that because I don't think it says here. So if we increase the frequency of our stimuli, meaning we put the stimuli closer together, we have what's called a wave or temporal summation. So this happens if two stimuli are received by a muscle in rapid succession. So again, increasing the frequency. So what happens here is muscle fibers don't have time to completely relax between the stimuli. So the twitches are going to increase in force with each stimulus. Okay, so what you have the first stimuli, contraction happening, it's starting to relax, but before it goes back to baseline, we have a second stimuli. Okay, so then you contract again, and the calcium released for the second stimulus stimulates more shortening. So you can have additional contraction, a little bit of relaxation, contraction again, a little bit of relaxation, contraction again. Okay, so you're going to keep building up tension or shortening the muscle more and more and more, contracting it more and more and more as time goes on. Again, because you're not allowing the muscle to relax all the way in between. Okay, so that again is wave or temporal summation. If the stimuli frequency increases even more, the muscle tension ultimately reaches near maximum. Okay, so what we end up is we have unfused or incomplete tetanus. Okay, so we have the muscle contracting, a little bit of relaxation, contracting, a little bit of relaxation, but eventually we reach this maximum muscle fiber recruitment, which means all the muscles in that area of our body are contracting. So even though we stimulate it again, we're not going to increase the tension at this point because this is the max tension that we can get. Okay, so each stimulus, we're going to go back and reach that max tension time after time after time. And that's unfused tetanus. Again, we increase the frequency of stimuli so that the muscles don't have enough time to fully relax. And if it gets to this top part where we have this plateau, that is unfused tetanus. We're at maximum muscle fiber recruitment. We've reached maximum tension, and we just have these little contractions and a little bit of relaxation happening between. If we can increase the stimuli even further, we have muscle tension reaching the complete maximum. Okay, if this happens, we have what's called fused tetanus. This happens when the contractions fuse into one smooth, sustained contraction plateau. Okay, in fused tetanus, you'll see the stimuli are so close together down here that there is no time for the muscle to relax at all. Okay, so the first contraction happens, there's not any relaxation, right? Because when we have incomplete tetanus, there's a little bit of relaxation. In this, we have no relaxation, so we contract until again we reach max contraction, and then we have this plateau at the top. Okay, this ultimately will lead to muscle fatigue. Okay, so that is increasing again the frequency of stimulation. So increasing the frequency of stimulation increases the strength or the tension. Okay. We can also increase the strength or tension by increasing the stimulus strength itself. Okay, so in these examples, all these stimuli would be the same strength, but we can increase the strength of stimuli to recruit more fibers. So if the stimulus is stronger, more muscle fibers are recruited, leading to a greater strength of contraction. Okay, so if we see here, these are, say, like different voltage of the stimuli. Okay, so we have just a small little voltage, greater, right? And then at a certain point, we have the highest voltage here. In our muscles, there is a threshold stimulus needed to create any sort of contraction. Okay, so any stimulus that doesn't reach that threshold will not produce a contraction. Once we reach that threshold, then as we increase the strength of the stimulus, we'll also increase the strength of our contraction, increase the tension of our muscle. Okay, and that's because every time we increase the strength of contraction, we are recruiting more and more muscle fibers, and the more muscle fibers we recruit, the stronger the contraction is going to be. Okay, this happens only up to a certain point where we reach the maximum stimulus. So at 7 voltage here, see we have excited all of the muscle fibers, so we have reached maximal contraction. Even if we continue to increase the strength of the stimulus beyond this, there's no more muscle fibers that we can activate, so we're not going to create any stronger of a contraction because we're already at maximum muscle fiber recruitment. Okay. So there's a few terms you're going to need to know. Subthreshold stimulus is a stimulus that doesn't reach this threshold, so we will not have contractions seen. A threshold stimulus is strong enough to recruit muscle fibers, so you will have some observable contractions. And then the maximal stimulus is the strongest stimulus you can have to reach that maximum muscle fiber recruitment and the maximum contractile force. Okay, all motor units have been recruited for the maximal stimulus. Even again, if we continue to increase the strength of the stimulus beyond that maximal stimulus, we can't see a greater contractile force because we've already recruited all the muscle fibers. Okay. So we can recruit more motor units depending on the strength of our stimulus. Recruitment works on what's known as the size principle. So motor units with the smallest muscle fibers are recruited first. And then we continue to recruit larger and larger fibers as the stimulus intensity increases, and the largest motor units are activated for only the most powerful contractions. So if we want a really strong contraction, only then we'll recruit the largest muscle fibers, largest motor units at that point. Okay, so smallest motor fibers are recruited first, then medium, and then largest ones as we want to increase our tension. Okay. Muscle units, motor units in our muscle are usually contracted asynchronously, which might seem kind of weird to you, but some fibers contract while others rest, and this just helps to prevent fatigue. Okay, so if we're trying to do something that is requiring a lot of motor units, we don't want to recruit them all at once, because if we do that, they'll all get tired at the same time. So we kind of switch off back and forth to allow some to rest while others are contracting. Okay. We also have something that is a constant, slightly contracted state of all muscle, which leads to what's known as the muscle tone. So we have just normal muscles that are always a little bit contracted to help stabilize joints, keep our posture, and things like this. And this is due to spinal reflexes. So groups of motor units are alternately activated in response to input from stretch receptors and muscles. So every once in a while, our muscles are just have a little bit of contraction to help maintain this muscle tone. Again, to help keep muscles firm, healthy, and ready to respond. So just kind of keeping them on their toes at all times. This also plays a big role in, again, posture, maintaining balance, as well as stabilizing our joints. Okay. And then going back to some different types of contractions. So we have our isotonic contractions, where muscles change length and move a load. Okay. And these can be concentric, where muscle shortens and does work. So if your biceps are contracting to pick up a book, that's a concentric isotonic contraction. An eccentric contraction is where the muscle lengthens and generates force. So if you lay a book down, the biceps will lengthen, and that's also technically generating a force. Okay, so that's an isotonic, where the muscle changes in length. Okay, so anytime you're doing a sit-up or a push-up, if you're pushing down, it would be concentric. Pushing back up where their biceps lengthen again, that would be eccentric. Okay. You also have those isometric contractions. The muscle is not changing length. Okay, and this means the load is greater than the maximum tension. So if you're holding something still, so you're holding a plank or you're holding a book out in front of you, you're not changing the length of your muscles, but you are still creating tension in those muscles to hold that position. Okay, and that's an isometric contraction. So the mechanical events are the same in either isotonic or isometric contractions, okay, but obviously the results are going to be different. Okay, so either you're generating a force and shortening your filaments, or you're just keeping, your force still. Okay. For contraction to happen, obviously we're going to need energy. ATP is the energy that is given to the muscle to move and detach cross bridges, pump calcium into the sarcoplasmic reticulum, and pump sodium and potassium with that sodium-potassium pump to maintain that resting membrane potential. So we need ATP for 3 different processes, and if we don't have energy, we can't do this. The muscle is going to stay in an excited and contracted state, which is going to what leads to rigor mortis. Okay, our available stores of ATP are depleted in about 4 to 6 seconds in our muscles. ATP is the only source of energy for the contractile activities, which means it needs to be regenerated very quickly in order for our muscles to continue working properly. Okay, and there's 3 different ways this happens. I'm not going to expect you to go into like crazy detail about these, but you have direct phosphorylation. So it's a coupled reaction of creatine phosphate and ADP. It does not use any oxygen, and it provides energy for about 15 seconds, okay? And it provides 1 ATP for a creatine molecule. We have an anaerobic pathway, which is glycolysis and lactic acid formation. So we use glucose. You don't need oxygen, so anaerobic means no oxygen. You produce 2 ATP per glucose and lactic acid. Provides about 30 to 40 seconds of energy. And then finally, the aerobic pathway uses oxygen. Aerobic means it needs oxygen. It gets energy from glucose, pyruvic acid, free fatty acids from adipose tissue, amino acids, a whole bunch of different things. It needs oxygen, but it produces 32 ATP per glucose as well as CO2 and H2O byproducts, and it gives you hours of energy. Okay, so those are the basic types of ATP generation, and they're going to be used in different circumstances. Okay, so if we have a short duration, high intensity exercise, first the muscle ATP that's stored in muscles will be used for first. Then you'll have direct phosphorylation ATP used and made. If we have extended 30 to 40 seconds, we have glycogen stored in muscle is broken down to glucose, which we use an anaerobic pathway to create ATP. So all 3 of those are used in just really short duration exercises. If we have a prolonged exercise, that's where we're going to use the aerobic pathway to create the ATP, and that's breakdown of several nutrient fuels, some different fats, stuff like that, to produce more ATP for prolonged exercise. So depending on the type of exercise, you can have different mechanisms at play. Okay? So that is the end of part 3 of chapter 9. If you have any questions, as always, please let me know.